Good evening, everybody. Thank you so much for being here. My name is Jacqueline Norris. I'm the Head of School and Dean of the Sydney School of Veterinary Science here at the University of Sydney. I want to start proceedings, of course, by acknowledging the country on which we are today. So I'd like to acknowledge and pay respect to our traditional custodians, the Gadigal people of the Eora Nation, and it's upon these ancestral lands that the University of Sydney is built. And as you see there from the picture, the Gadigal people are from as far west as the Nepean River, so our other campus is just over the other side, down to the Georges River and up as high as the Hawkesbury River. So as we share our knowledge, teaching, learnings and research practices within this university, may we also respect and pay our respects to the knowledge embedded forever within Aboriginal custodianship of country. So this is the first of two beautiful paintings. This one is by Rebecca Beatson called The Joining of the Two. And I guess I chose that one particularly because our special guest this evening, of course, is from our sister country, New Zealand, the country a sixth the size of us, but is always 100 years ahead. I think we'd all agree with that, with most things, particularly their respect for their Indigenous culture. So tonight, this is the Ian Beveridge Memorial Lecture. This is the second one for this year because the first one in the year was actually a very late 2023 one. So we have it every year, and it's to acknowledge the work that Ian Beveridge did in conceptualising the concept of what was then called One Medicine. So I've got a kind of framework in the middle there of actually what our students are taught now, which is One Health, One Welfare, managing those shared risks and maximising wellbeing. And if I think about the kind of work that Ian Beveridge did, he was a graduate actually of our veterinary school in 1931, and the ideas that he came forward with at that time within our profession and across into medicine in that framework were certainly ahead of the time in this particular area. So he obviously had major advances in sheath rot in sheep, etiological agent causing foot rot in sheep, influenza viruses. He looked at international nomenclature and classification of cancers of domestic animals, and then he went and established the Cambridge Veterinary School and was chairman for 18 years on the World Veterinary Association. So he had a very big influence on the way in which people started to think about the interconnectivity between animals and people, and then of course people and the environment. But just to be, and certainly this lecture is to acknowledge that, but just to be incredibly clear, our Indigenous people were thinking about One Health, One Welfare for a very long time. It's present in their art, it's present in the way they care for country and the way they interconnect all aspects of life. And to connect our speaker tonight to our Indigenous culture, I came across this wonderful Maori expression of kamua kamuri, I hope I'm saying that correctly, which is to walk backwards into our future. So to really think about what our future might look like by actually stepping ourselves back into the past and reflecting that kind of history through. So now it's my great pleasure to introduce Jackie Benschop. So Jackie, of course, is a veterinarian, worked in clinical practice and the meat industry in New Zealand and the UK for 15 years, and is now a co-director in the Molecular Epidemiology and Public Health Laboratory at Tuahara Ora, the School of Veterinary Science, Massey University. And her work is really centred around interfaces, ethics, One Health, equity, diversity, communication. And I was very lucky to have to write something about Jackie for a different context previously, and it gave me an opportunity to really understand what she had done. And these are some of the things that I wrote about her, that undoubtedly a global leader in zoonotic diseases, with her work focusing on the diseases that have major occupational or foodborne transmission cycles that have significant outcomes for populations locally and globally, and that her drive to improve the lives of others fuels these endeavours. And that her work, although she's going to talk about leptospirosis this evening, her work obviously goes across many other of my favourite bugs like Campylobacter, antimicrobial resistance, and a whole lot of other things. But I think the thing that really differentiates Jackie is her purpose, the way in which she brings that incredible light and brightness and collegiality to everything she does, and actually wants to improve the lives of others by the way in which she does research. So it's my great pleasure to welcome Jackie.
It's a great pleasure to be here. It's the first time I've been at this campus and able to spend time in this lovely city, and I want to talk with you about leptospirosis. As Jackie said, I'm working at MEPI Tawharo Ora in the School of Veterinary Sciences. I also want to mihi to One Health Aotearoa, a research consortium, the Global Leptospirosis Network. We spend quite a lot of time in support of our outbreaks in countries, including protocols and capacity building, and our group is a collaborating centre for the World Organisation for Animal Health. I want to acknowledge the Health Research Council of New Zealand. They're not directly funding the small piece of work I'm going to talk to you about, but they support and have supported my team for many years. I want to follow in Jackie's footsteps and acknowledge the Gadigal people of Eora Nation, who are traditional custodians of the land on which we meet tonight, and pay my respects to their elders past and present, extending that respect to Aboriginal and Torres Strait Islander peoples here tonight. I want to mihi and greet you in Te Reo Māori, which I'm a novice in, the language of the Indigenous people of Aotearoa, New Zealand. Tēnā hui hui mā tātou katoa ko Horani in I tō nihia me inga rangi o ko whakapaparanga mai. Ko Bessie Rao, ko Henriette, ko Oko Kuia, ko Fred Rao, ko Jan William, o ko Kuro. Ko Jackie toko mama, ko Case toko papa, ko Mary toko tuakana, ko John toko tunane, ko Maria toko teina, ko Jackie Benschop toko ingoa no o Tatahi a ho, ko Oscar Rato ko Tess, ko Ben, ko Trina aku tamariki, ko o Tatahi toko whenua tupu, ke pai kakariki ai noho ana ko Ngāti toa te mana whenua. Nō reira, tēnā koutou, tēnā tātou katoa. Just as reflected by Jackie, I'll start at the bottom of this in acknowledging the land on which I come from. I come from Christchurch. I now live in Paikakariki, looking out over Kapiti Island, across the Tasman Sea towards you here. Ngāti toa are the local iwi or tribe that look after the land. And you have there my whakapapa, my grandmothers, Bessie and Henriette. There's a picture there of Henriette, my grandmother, with my father Case, my sisters Mary and Maria, my brother John, acknowledging the role that these people bring to me and the lands in which I work. My children, Oscar, Tess, Ben and Katrina. Ki te kotahi, te kahaho, ka whati, ki te ka puia e kore e whati. This is a Māori whakatauki or a proverb, and I try and use as many of these as I can. In the middle is a strong bunch of reeds. This is our staff that work together on the pathogen system of leptospirosis, and we are complex, I suppose, in our diversity, in that we have people from social work, that's Polly, micromolecular biology, Shah, Stu and Julie, Gerard from social science, informatics, Ahmed, myself, veterinary science, and surrounding us are some of our students from over the last 10 years. And I don't have to remind any of you, I'm sure, how valuable and what wonderful work our students do and the life they bring to our work. Jackie's already presented a model of One Health. Just remember that the Beveridge Memorial Lecture is around One Health research. This is the definition I'm going to talk to, but more particularly a definition, the third part of it, which I find the trickiest part to bring to my research. One Health is integrated and unifying, is looking to sustainably balance and optimize the health of people, animals, and ecosystems, recognizing the linkage and interdependence. I think we're comfortable with that. We can think about that. This is where I find it gets tricky, or particularly tricky. The mobilization of multiple sectors, disciplines and communities at varying levels of society. So if I think about leptospirosis as a pathogen system, and I take one example, say around the meat industry, and the different levels of society at which we engage with the meat industry, that's both at the level of the industry body, the Meat Industry Association, the different companies, the health and safety people, and then also the Meat Workers Union, who have also supported our work. All across that industry, we've had tremendous support. Similarly, with different sectors or communities within human health, nurse and general practitioners and occupational physicians we work with, but also the public health and Māori health people working at the district level, and then also centrally, people in the Ministry of Health. That's the first part of part 3. The second part of course, those larger global issues that now more than ever we need to focus on in whatever we do, and even working in what might appear to be a narrow sphere of infectious disease and a number of pathogen systems, always having an eye on the bigger picture about global health, clean water, and climate change. Here's an image of that, which is similar to the image that Jackie showed before, and to your, let me get this right, to your, the sectors and disciplines, and the One Health here. If I think about myself, primarily veterinary trained, I should be fairly comfortable in that sort of healthy animal sector space, and increasingly comfortable perhaps with healthy humans and human sectors and disciplines. I know as a research group we're working as hard as we can to take on board and think about the environment and the ecosystem and the health of it itself, rather than the environment as just a conduit of pathogens between humans and animals. Here are the 4 cogs that make this happen: communication, collaboration, capacity building, and coordination. But that circle called society, I find that the tricky one, and as a One Health researcher, I want to be able to be inclusive and think about equity and access and the scale at which we work. This is the definition from the One Health high-level expert panel, the quadripartite group that are working during and since COVID on both defining and operationalising One Health. And I want to mihi here to Dave Heyman, who's the co-director of the lab in which I work, and he's also a member of the One Health high-level expert panel. Another model, I quite like models for complex systems. I want to thank Jared Princeton for this one. This is about transdisciplinarity. I think One Health by itself is transdisciplinarity, and I'd be happy to hear you say to me, well, what do you mean by that? I'm going to try and explain it by this particular model, where in fact if we start with the x-axis here of this particular image, we move from high to low engagement of disciplines. So if I think about my university and engaging with disciplines of veterinary science, of social work, of psychology, of ecosystem health, agriculture, and so on, and the closer we stand together, the more we become interdisciplinary. But that's just us as academics. I do quite like this model, because as we move up on the y-axis here, we start to think about being more participatory and starting to engage with those that are not academics. And I've talked already about a couple of groups, those that may be in medicine, those that may be in the meat industry. We want to really be in a space where we are both high in the engagement with disciplines and high in the engagement with communities and stakeholders. And if I can, I'd like to show you an example of that in some of our work. It's not just, this is not a new idea. In fact, Ian Beveridge, in The Art of Scientific Investigation, I found some summaries that Ian Beveridge had written about some of his chapters, and here's one that I've used to say speaks to being transdisciplinary. From Ian Beveridge, once we've contemplated a set of data, the mind tends to follow the same line of thought each time, and therefore unprofitable lines of thought tend to be repeated. I know that happens to me. There are two aids to freeing our thought from this conditioning. One, to abandon the problem temporarily, and that helps as long as it's temporary. Secondly, to discuss it with another person, preferably someone not familiar with our work. I like to see that as being transdisciplinary. One thing I must remember, and a lot of my work now is based in Aotearoa, New Zealand, that as a treaty partner and a priority population, part of me being in this space is to think very deeply about Māori involvement. Here's another model system. I finally got to talk to you about one of my favourite pathogen systems. This is an image from Nature Reviews Microbiology about leptospirosis. To your right-hand side is the person or the patient with leptospirosis. And you can see that the presentation can be very complex. You do need diagnostic tests to diagnose lepto because it can look like many, many other things, particularly in settings where there's multiple other causes of acute febrile illness. Just to keep thinking about this person though, if we go a little bit deeper, we were lucky enough to do a case control study of leptospirosis. Unfortunately, it happened to be over the time of COVID, but we still managed to enroll 95 patients with leptospirosis and 300 controls. We followed up those 95 patients between 8 and 18 months later, on average about a year later after they first became ill, and half of those patients still reported having at least one symptom. Many had had to change jobs or modify the way they lived their lives. So yes, it's a severe acute disease, but it has a long tail and a long recovery. Other things about that person on the right-hand side, at least in where I live and work, this person likely has some insecurity perhaps, or some challenge in their employment. This person is often a farm worker or a meat worker that may be relying on their employer for their meat, milk and home, or perhaps is working in shift work. Perhaps they only have 5 days sick leave, so they therefore can't take a lot of time off work. So there's issues around agency and power in the people who contract leptospirosis, and of course not only leptospirosis. The middle segment here is about the environment. At this stage it's shown as a conduit for pathogens, and I said I wanted to move away from that model, but at least for now it shows us that when people get leptospirosis from an animal source, they can get it directly or it can be indirect through soil or water. We'll talk a bit more about that in a minute. Whoops, and I haven't talked about the most complex of all. To your left, the mammalian host. So here's at the top, asymptomatic rodent carriers. In this model, this is proposed as the most common global model for leptospirosis. But I just would like to mention that there are many other asymptomatic carriers of the bacteria Leptospira, particularly cattle, for example, but not only cattle, many other wild animals and domestic species as well. It's a very complex and interesting interaction between the pathogen and the mammalian animal host, where there's a sort of soft immune response and the bacteria is not cleared. This sets up a situation where the bacteria are sitting quite comfortably in the renal tubules, and then there's intermittent shedding of the bacteria in the urine. And that is how people get infected, particularly through grazes and cuts on their skin, perhaps when they've got soft skin from wandering through wet conditions, or also through mucous membranes. Animals can of course get leptospirosis too, but I'm not going to talk about that tonight. It's a global disease, absolutely, whichever way you measure leptospirosis, and these are always under ascertained, these counts, whether it's by disability adjusted life years, deaths, or loss of human productivity. This will always be under ascertained. I know that's not peculiar to leptospirosis, but burdensome, without even considering the burden when we have loss of animal growth, abortion in animals, loss of production in animals, whether it's in a complex farming system or in a subsistence setting where a family has just a few sheep and goats, and there's then loss through abortion. So sometimes you have the worker taken out in the family, workers, people sick in the family, and also their animals in a subsistence setting. I mentioned it's bacterial infection of mammals, and here's an electron micrograph. It's not hard to imagine. It's a very fascinating bacteria, Leptospira. It's a spirochete. The motility and how this bacteria moves is phenomenal, really, and it's not hard to imagine it burrowing in through a mucous membrane and setting up an infection. When I first started looking at lepto about 15 years ago, there were around about 18 pathogenic species. Now we're, of course, in the era of genomics. Many more have been discovered. There are, of course, saprophytic lepto as well, but our interest is in those that cause disease, and it's been described as a blooming tree because generally the more that's looked for, the more that is found. There's a more traditional approach, non-genomic way of looking at Leptospira as well by looking at serology, which is used both for distinguishing lepto and also for diagnosis. And if we think about how it's set up for people, what we see globally, there are 3 patterns. Poverty. So poverty perhaps associated with living where there may be rodents, for example, or having poor flooring. Flooding. Flooding is really interesting. Not only when there's an initial flood or extreme event, animals and people come together and seek high ground. Therefore, there's more chance for contact. But also with flooding, animals are stressed, they may be shedding more. People are stressed, they may be more susceptible. People have macerated or wet skin that may allow leptospires in as well. And then what we tend to do with flooding, of course, is manage the debris afterwards, whether it's material from flooded homes, whether it's flooded orchards and all the trees and apples, or whether it's dead stock. We tend to bring the rubbish together and then create another environment where rodents and say other wild creatures can find homes and mates and food. And we often get a second wave about 3 to 4 weeks after the flood. And then occupational. This is what we tend to see in Aotearoa, New Zealand, but that's not only what we see. So we've called it their dairy farm fever, cane cutter's disease. Anywhere where people are working and they may have a wet environment or be working with animals. What about the soil? Very interesting to me is that over the last 4 or 5 years there has been a heap of literature around the role of the soil in leptospirosis, and discussions about soil acting as a reservoir or at least a temporary carrier of lepto. So I've already talked about floods in one light. There's another way to think about floods is, of course, dispersing bacteria and increasing the risk of exposure by disrupting soil structure. So disrupted soil structure can be, of course, from just water coming up, but particularly near where I live, where we have a lot of bare soils or bare pasture and a lot of waterlogged soils, and then slips. We get slips of soil into waterways, and Leptospira has many interesting trades. One of them is forming biofilms, and then biofilms can be quite stable in soil, and then disrupted soil can release them. And Leptospires generally are maintaining their virulence after they've been in a biofilm. And this is just another way to think about the effect of climate in Leptospira. Some pictures from colleagues overseas in terms of potential ways in which people may be exposed. It's interesting, it's not just in wet areas. I've mainly shown you pictures of wet areas here in different parts of the world, but there are a couple of dry spots too. And I just want to draw your attention to the bottom right. This is a work done by colleagues in the Institute Pasteur in West Africa, in Niger. Very arid place where a very good idea and an appropriate approach to bring food into the cities is to start greening the areas around the cities with irrigation. And I guess it just shows you, or it makes me think about the dynamic nature of infectious diseases, that once a new environment like that is set up, where there's irrigation and greening and moisture in an otherwise arid setting, that there are hosts and pathogens, endemic rodent hosts and particular strains of lepto that are now set up there. In the middle at the top, poor living conditions in Sao Paulo in Brazil, open waterways, harbourage for rodents, and open floors. And Federica Costo and Albert Coe have done a lot of work looking particularly at rodent-borne leptospirosis in that setting. And then the picture to the top right, humans and animals post-flood in Hawke's Bay in New Zealand. Moving now to the situation in New Zealand, I talked about there being 42 species of pathogenic Leptospira. To the best of our knowledge, this is what we have circulating, both based on originally on culture and now on genomic methods of detection. 2 species, Bull petersoni and interrogans. And if you look at the ones I've put there in bold, Cerevars bellum and Harjo and Pomona, these are the ones that we most commonly see in human cases. The ones there with the red asterisks, Harjo, Tarasovi, Pomona and Copenhagen, we have livestock vaccinations for these Cerevars, which are actually very well engaged with in the dairy and pig industry. And if a herd or a group of animals is vaccinated every year and at the right time and young enough and the cold chain is maintained and so on and so forth, these vaccines are very, very efficacious at actually eliminating shedding from a group of animals over time for those strains. Unfortunately, there's little cross protection in these vaccinations. The drivers for vaccination in the dairy industry are as strong for human health as they are for cattle health. And the pig industry, similarly, in fact, commercial pig herds in Aotearoa, New Zealand, need to have a leptospirosis reduction program in place before their pigs can be accepted at abattoir. There's a rough grouping. There's nothing exact here, of course. The maintenance host can itself become ill with that strain if things aren't going so well. Perhaps there's times of stress or what have you. But nevertheless, we feel this is the best model for what we see in Aotearoa. And if we look there, we tend to have a bit of a split in terms of our wild animals. All of our wild animals, or rodents, hedgehogs, possums, we have 3 species of rattus and just the domestic mouse, Mus musculus. These are all considered pests in Aotearoa, New Zealand. So you might hear me slip into the use of that word, pests, for these small free-living mammals. They tend to be considered as maintenance hosts for the strains Bellum, Balcanica, Tarasovi and Copenhageni, whereas our livestock animals are maintenance hosts for Harjo and Pomona and the strain Pacifica. That's a rule that helps us a little bit with source attribution, and it's tended to be stable over years of studies. I want to move now to one specific example, one thing that's quite local as opposed to global. I only saw this paper a couple of nights ago. It's just been published, and man, it just struck me. I thought I would use it to introduce Cyclone Gabriel, because I want to talk about that as an event which I'd like to show you how I feel we work towards being truly working in a One Health and transdisciplinary way. But the reason that this paper talked to me was, for the first time, and I haven't been looking at this literature, but for the first time it talks about the anthropogenic warming and how it can increase the amount of rain delivered by this type of storm by about 10% per degree, and similarly increase the peak amount for this type of storm by about 20 to 30% per degree Celsius. That's an aside, but it just struck me, particularly at the moment. Why I'm showing you this is to introduce Cyclone Gabriel, which passed along the northern coast of Aotearoa in February 2023, produced historical rainfall accumulations and impacts, loss of 12 human lives, many animal lives, loss of infrastructure. It was an ex-tropical cyclone that stalled and re-energized off the north coast. resembling descriptions of worst-case scenarios for the northeast of the country. For me, the most frightening event in my lifetime of this nature, which I know makes me lucky. It's really nice to have the big screens so you can see here the areas affected by Cyclone Gabriel across those 2 days. I want to zone in one particular area. Hawke's Bay. Don't know if many of you have been to Hawke's Bay. It's a lovely area. It has 2 quite small cities, Napier and Hastings, cities of around 40 to 60,000 people in each. Predominantly a fruit bowl and orcharding area, sheep and beef cattle farming towards the hills, a little bit of dairy farming in the south. Population of Hawke's Bay is proportionately more Māori than the average in New Zealand, around 30 to 35% compared to around 20%. And this impact layers of these maps, as shown in blue, show inundation with floodwater, deposited silt and bare soil on steep slopes. This area, a bit like my home area of Canterbury, has a large number of braided rivers. These had high stock banks with cycle tracks along the top. And there was breach of stock banks, and particularly frighteningly affected when rivers either side and breaching either side. And the silt is a really terrible picture, but shows some of the depth of the silt that comes from these rivers, and of course there would be tremendous loss of life in livestock. This was one lucky cow that managed to survive and be rescued. So a very impactful event. Within 1 week of this event, we were contacted by Naira Haka, who's a senior workforce manager of Te Aka Whaiora, the Māori Health Authority in Hawke's Bay. And we've been lucky enough to be working with Naira and Māori Health in Hawke's Bay for a long time before then. And she said to us, we're concerned about diseases from animals, not only lepto. Together with others from public health in the region, we came up with quite a bit of information that was shared as well as it could be, both in Te Reo Māori and Te Reo Pākehā. This one particularly about lepto, but also about salmonellosis, for example, and just about general hygiene and concerns with floodwater. So the speed at which this material was put together was, I think, quite remarkable, and I thought it quite, there was quite a lot of foresight, I think, for people to approach us, to ask us particularly about this. Many people were cut off, and there were many people working and clearing floodwater. And these messages were delivered by social media and haora, so Māori health centres as well, but of course many people could not get to these centres. I want to talk about how come I think we were asked and advised so quickly and asked to help so quickly. I've been really lucky to be working closely with the Māori Relationship Board in Hawke's Bay. This is a board of Māori that works with what used to be the District Health Board. It's still kind of a district health board, but it has a slightly different name. And some work that Shanisa did in looking at the table where she reviewed cases, notified cases over the last 20 years or so, show that compared to non-Māori, Māori are about 1.3 times more likely to be notified with leptospirosis. So they've got through the pathways of being sick, having a sample taken, having a positive test, and so on. But Māori are particularly 2.7 times more likely to be diagnosed with pomona, which is a much more severe type of lepto generally than the others. Now, I'm not showing you the data, but on the back of our case control study of 95 cases, Māori were also 4 times more likely to be hospitalised than non-Māori and have a longer hospital stay, even though on average they presented about a day earlier with their condition. So we started to talk earlier on about inclusivity, equity and access, and this is just one example, one disease and one example, but there are many in my country of how a particular sector of society has, in this case, poor health. Another reason to think about, and Naira showed me this photo, there are many Māori exposed to leptospirosis in Aotearoa, New Zealand. And this is a picture of Māori at now a closed meat plant. many family members working there, and also there are many Māori working in farming. In fact, Naira herself, her uncle had lepto, and she said there are many whānau was Māori for family living with lepto or long-term lepto, what they felt was long-term lepto in Hawke's Bay. So what's in blue is when Naira presented about our relationship to the District Health Board, and she's happy for me, obviously, to share this with you. So when we first met Jackie and the team, and this was about 2016, the aim was to ensure they felt welcome. And that was wonderful. In fact, in terms of feeling welcome, the meeting was kind of, you know, around after lunchtime, and I thought, well, we better have lunch first. But that was a mistake, because of course part of the welcome was that we have some kai, that we have some food. Quickly on from that, we were there to look at how this research would benefit our population. No question, really direct and punchy. And I like that. How will what you do benefit our population? That's quite different than turning up and saying, oh, we'd like to do this work. It's a slight different angle, and I think that it can present some challenges. Our early meetings were initial whanaungatanga. Now this one doesn't really have a direct English translation, but this is about getting to know you, becoming a family. We had many meetings before we really got to talk much about what we wanted to do. Who are you? Where are you from? Can we trust you? Because there's history of distrust of people like me in my country because of the colonial history of Aotearoa. Naira said we got to present who we were, our goals and team and aims as tangata whenua, that's people of the land. She presented, that's me, she presented her team's goals. From there we formalised a trusting relationship. And JB Perry-Smith there, he himself is a CEO of Ngāti Kahungunu. So Ngāti Kahungunu Iwi is the tribe up there that works at the Māori Relationship Board, that works with the District Health Board. JB himself had worked in the meat industry and knew about lepto. And this is what happened after the flood. So on the x-axis are weeks from 1 to 52, and this is 10 years of data. The blue line that looks different to all the other lines is what happened in 2023. On average, there's about every year, the last 10 years, 8 to 10 cases of leptospirosis notified in Hawke's Bay. In the year after of the flood, there was close to 40. There's a few things to note here, of course. For a start, it was a wet summer, so notifications at the start of the year were already up. The cyclone was on the 12th of February, and a very steep curve of cases, almost 20, within that 4 to 6 weeks after the cyclone. So definitely there was an increase in leptospirosis. It looks like it was associated with this extreme weather event. Together with Māori health and public health, both of Hawke's Bay, but also Te Tairāwhiti, the DHB that's above, another east coast, we wondered if we could do some work together about this increase in cases. Because the very neat thing about working with people in human health, particularly medical officers of health, is they are very keen on epidemiology and really keen to talk about zoonoses with us. And we've got a history of that. This was our hypothesis. Not stunning, really, given what we know globally, but we thought, gosh, I wonder what kind of lepto people are getting after the floods. And our hypothesis was that it would be associated with rodents. And together with public health and Māori health, we applied for local funding to use strain typing to try and understand what was happening following these extreme weather events. Just one thing about doing research in a community that's been devastated by a flood is, even though there's many things you might like to do, I think quite hard about the ethics of what we should do given all of the complexities and recovery that this community needs to go through. What we decided to do, and I just want to acknowledge here the master student at the bottom of the text here, Preeti Pandey, who did this work, and thank the Canterbury Health Laboratories and Waikato Hospital Laboratories for the supply of genomic DNA. So we wanted as much as possible, and this was advice from NIRA, from Māori Health, and from Public Health, to use what might be available as opposed to disrupting a community in recovery. There's only two, in New Zealand, there's only two diagnostic human laboratories that run PCR. That's the Canterbury Health Laboratory and the Waikato Hospital Laboratory. And we asked them if they could hold the genomic DNA from those human samples, pending us, of course, getting ethics for that and also getting funding. And they did, and what we were able to do was to use our research PCR, which doesn't just say lepto yes or no, but allows us to look particularly at the GLMU amplicon that we use to determine which particular strain of lepto there is. We were able to do this for around about 1/3 of the diagnosed cases of lepto that year. What we found here, and we have here month, this is just 2023, and looking at March, which was immediately after the cyclone, we have a mix of both livestock and rodent-associated cases there. The top three colours, Balkanica bellum and Copenhageni, are more wildlife or pest-associated, rodent, possum associated, or mouse, whereas Harjo pacifica and Pomona are livestock associated. So yes, there was an increase in cases in March, no surprises there. We were most interested in Hawke's Bay. Here we have the different health regions alphabetically along the bottom here, from Auckland to the West Coast. And when we look at Hawke's Bay, it's really interesting. I acknowledge that numbers are low. We have 13 cases there, and 12 of them are actually livestock associated. So that's always really refreshing when you have an idea and you think, oh, I don't think that's what's going on, at least in Hawke's Bay. Certainly there seem to be many more livestock associated cases. Now, after that, this was a really interesting case, a situation where humans were sentinels for animals. Because at the time when the human cases were increasing, there was no reports of increased cases in animals. And we know why that is. The way veterinarians diagnose, and there was animals stranded, farmers were stranded. But over time, we became aware anecdotally, and then we also have data to show that there was a lot more cases of leptospirosis in sheep. And Hawke's Bay is a predominantly beef, cattle and sheep area, and these animals are not vaccinated. So, information we're starting to share with some caveats, and we obviously want to publish and so on, is that at least post-flood, livestock-associated Pomona predominated. We know Pomona causes a more severe disease because it's an interrogans as opposed to a Ball-Petersoni. We do want to keep giving a message that both livestock and rodent sources are important. And the bottom line is that the risks to animal and human health will reduce if beef cattle and sheep and deer are vaccinated in a full vaccine program. And I know that's a hard ask, particularly for New Zealand farmers, but when we had a rodent hypothesis and we have now a domestic animal source, there's a pretty clear message to actually say, hang on a minute, we actually do have some tools here that we can use. Whether that's practical or not, of course, that's another question. Coming up on the 25th of November is something pretty exciting, kind of unrelated to the cyclone. So this piece of work, preparing information by Māori and for Māori, is funded by a larger Health Research Council grant, which has nothing to do with the cyclone. But it happens to be focused in Hawke's Bay because we have a lot of history there. So we're having a hui, which is a meeting, which is on the marae, which is prioritising and talking about lepto in a Māori space and in a Māori way. And we have been working with Kailani and her creative company, Pure, which actually means play, like playing a game. It's a creative company. She's done work with health before. And yet to be finalised, but there will be designs that are specific and have particular significance culturally to Māori, will be translated into Te Reo Māori, and she's also created a lepto game. You get 6 points for covering your wound. Her idea is, of course, that we release this to tamariki, to children at school in Kahangareo, so Māori immersion school, and that the children are playing this game and talking to their family about lepto. Wish me luck for that. I want to finish soon. I'm not sure of time, about 5 minutes. Yep, thank you. I really appreciate being, or trying to be, transdisciplinary. I think it gives me and my team an ability to be much more comfortable with tension because we're in an uncomfortable space. We have to make ourselves quite vulnerable about things we don't know, whether it's a discipline or whether it's a culture. We have deep and unexpected understandings and many new research questions come up. So I've been really fortunate that the 3 big research questions of the current Health Research Council grant we got all came from the community, whether it was a Māori community, a medical lab community, or public health, or rural women, a community group. And it's personally, I find it very satisfying. The view that I have of my world is just one view, and I like to learn about that difference. It doesn't always work though. Well, I think it always works, but it's not easy, right? As soon as you introduce difference and vulnerability, it gets hard sometimes. And even across disciplines, even with colleagues in social science, we have to be open and vulnerable to learning new terms. I think we have similar values, but different approaches to questions. It can be really hard to engage. I don't know how it is for you, but for me, engaging with Indigenous people is a long-term, long game, and there are many, many calls on Māori in New Zealand to help and be involved with work. Mine is just one of them. And because of the size of teams and because of the complexity of people, I spend a lot of time on communication, which means there's many things I haven't done, many skills I haven't been able to develop. I'm okay with that, but I need to be comfortable and weigh that up. And I also have some discomfort, just because of some of the things I'm exposed to and see, and some of the history of my country. If you want to read about that, this is a really good book called This Pākehā Life, and I'm sure you may well have books like this in Australia. Conflict does occur. This is an opportunity for growth. We're starting to engage with a new community in North Auckland, which has asked us to do some work. And so one of my PhD students has been looking particularly at how we should engage with communities and doing this the right way around and looking at literature. And what she said to me about research priorities may differ. These are the 2 big things she said. The other thing was about communication. Often researchers won't get back in time at all. Well, ideally what we're doing is we're actually developing our results together with the communities we work with. But this last point, community partners may oppose dissemination of findings that could cause harm to the community. This is a very recent example, and it's been very conflicty. So we published this paper very recently. The reason why I got it, messages from people diagnosed with leptospirosis about infection in the workplace and its impact on livelihoods. This came from our case control study where we had 95 people with lepto and we did surveys of them and 300 controls. We also interviewed in detail, face-to-face, 13 people with lepto to capture experiences. And there are some things in our publication that we knew would be touch points and concerning for our partners. For example, people know about personal protective gear, but they remove it or take shortcuts knowingly, often under time pressure. For example, meat processing workers felt forced to return to work too soon because they needed the income. For example, ACC, which is our global national no-fault workplace insurer, people felt disrespected and dispirited by their association. Now, we work closely with the meat industry, with ACC and with many other groups. And this is only the abstract of the paper. There was a lot more detail that we knew would be concerning and not shed good light on these communities. The approach we took, I don't know if it was the right one, we asked these communities if they wished to provide commentary on our findings and actually have that co-published with us. Only one group did. One of the groups that didn't had these comments for me. The negative messages about processes could be potentially damaging in the press. These responses don't reflect the care and concern we have both seen, both me and them, from employers who want to avoid the disease and help people. It does the majority a disservice to publish a report that says employers aren't caring or supportive enough. This was a challenging time. We had a number of meetings with particularly one particular industry group, and we've published the paper. We didn't make a press release. I feel with my hand on my heart that our relationship with this industry group has actually grown through going through this process. They could see that these messages were genuinely there, and we could see their point of view. I don't feel that conflict has been harmful for us as yet. I want to finish now. You might recall this circle about One Health, the one I thought was difficult, and I still think is difficult, the one about society. One thing I found really useful is this particular table here about claiming space for multicultural transdisciplinary research. And there's some guidelines here, 7 guidelines. And they're based on Māori cultural values, which are I think I can safely say are no different from the cultural values that many of us hold without doing a disservice to anybody by saying that. About respect, about looking and listening and then maybe speaking, about being generous, being cautious, do not trample on the mana or dignity of a person, and particularly this last one. Himahaki, be humble, do not flaunt your knowledge, find ways of sharing it. Nō reira, tēnā koutou, tēnā tātou katoa.
Thank you so much, Jackie. That was just fascinating. The immersion of your work amongst every part of all ecosystems within New Zealand in what is really quite an interesting stealth pathogen, but then emerging as really quite a. I do have a question, but I'd like to ask the audience if they've got a question. Cass has got some microphones rolling. Fantastic, thanks.
Thank you. Is there a vaccine in the works for humans? And do you have a clinic that helps people with long lepto?
Thanks very much for that question. There are human vaccines available. If I was a sewer worker in Paris, which I'm not, I'd be offered or be actually required to be vaccinated against a Copenhagenized strain. The problem with the vaccines is that there's a lot of, there's no cross-immunity between strains, and other countries, Cuba and China, have also had human leptospirosis vaccinations. I think that the reason, there's a push of work and interest, but generally lepto is affecting people in poorer aspects of the country. There's certainly not one available in New Zealand or Australia. Your second question, thanks so much for your interest. I've got a lot of interest in what I'm calling, I'm calling long lepto, or the long tail of recovery. And we have a grant in at the moment with the Lotteries Health to look at this cohort of people for the first time globally, people who have had lepto, and to follow them through and see how they go. At the moment, there is no community of support for those people, but I have written that into the proposal, and I'm keen to talk to you later about that. Thank you.
Thank you for a very educational lecture. James Thompson from Turramurra Vets. Do you have much in the way of urban cycles of lepto in New Zealand? Because we've had a flare-up in Sydney recently.
Well, thanks very much for your question. We're currently looking at an urban community, a coastal community, so it's not quite urban, in Auckland that was affected by floods and has a freshwater lake that we've detected lepto in. I think the short answer is no, but we haven't looked. And when I talk to Naira or people in Māori health, we talk quite a bit about, say, rodents, sympatric rodents in urban centres. This is something that we haven't looked at, but it's been well looked at, say, in Brazil, and I know a little bit about the case in history, the history in Sydney, sorry, and I think it will be there. Does that answer your question? Yep, thank you.
Thanks very much, Jackie, for a great talk. I'm really interested in these guidelines that you have on the slide there, and in particular thinking about sudden events, kind of disaster events, for example the flooding in Hawke's Bay, but also more recently the COVID outbreak, and the kind of pushback on control interventions that public health authorities might generate for these events. Are there any kind of specific points of learning that we can take forward to maybe try and engage with the public a little bit more widely as an epidemiology community that would actually help when we have to put in interventions to deal with those kind of natural disease disasters?
Yeah, thanks, Chris. I think for me, it's being known there, and I don't have to be, or we don't have to be, world leaders. We just have to be the best that we can be and be known and be ourselves and understand that there's an understandable distrust around research. There's a very lovely example about COVID, though, in Aotearoa. I mentioned something called Te Aka Whaiora, the Māori Health Authority. Unfortunately, that's been disbanded. But at the time of COVID, it was that group that was able to have the trust and work and have mobile vaccine, trained a lot of vaccinators, had mobile vaccine sites, and also turned up at Marae and vaccinated. So yes, these guidelines, which are fine, right, because it's peacetime, I guess it's doing that preparatory work with the relationships. I think having somebody turn to you, as we did, after that flood, that really brought it home to me, because I knew that we had quite a good relationship and so on, but people are busy, they have so many complex things, and zoonoses really aren't top of public health, understandably. There's so many, many other competing things. But just because we were there and known and trusted, it was very nice to be called upon. Thanks, Chris.
It's just one. Yep.
There you go. Thank you. Thanks, Jackie. Sorry, it's just a quick, boring technical question. I'm Mark from the State Veterinary Lab. We want to develop a PCR to do serovar testing, and I wasn't aware that there was one available, so just it wasn't whole genome sequencing of, it was a primer-specific assay, was it? And where are the details that I could grab that from? Is it published?
Yep. Thanks for that question. Only works in New Zealand, sorry, so that's what I mean by a local disease. So we have a relatively small range of serovars. So yes, it's a specific amplicon sequencing of the GLMU loci that differentiates strains in New Zealand. but it won't necessarily work across global strains. But yes, it's published. Wilkinson, I can make that available absolutely in PLOS One. But I know I had the title a global disease, but a local phenomenon. But I thought I want to talk. I didn't talk too much about that. But one of the things, of course, is locally appropriate detection and diagnostic tools. When you have such an importance around what's happening, what's circulating, and what are your hosts. Yep. Thank you.
I was going to ask the same question, Mark. Mark Westman works with us, and the emerging thing in New South Wales, and to James's question as well, is started all in dogs with Copenhagenii, and then the more that we've looked, and unfortunately at the moment we just have microagglutination tests, antibody tests, which of course have its failings, as we've been the people that follow these dogs through, the number of other serovars, because we do it through the World Health Organization lab in Queensland, we've now seen it's actually shifting more towards Australis and some other strains, all within dogs, and so it's really like an emerging question. It's like the other question I had for you was around when you had the theory that it was rodents with the cyclone, now you've got Harjo and Pomona, which, as you said, is livestock associated. What's your thought processes around, where those cases occur and the geo-mapping of it, and that makes sense to you now because of the animals in that area? Or, and I guess in my small brain I kind of think, well, what happened? I mean, the cyclone landed all this water and salt water there, and what shifted to have so many cases in that area like that?
Yeah, that's really interesting. Emily Valet did some excellent work on cases reported by our veterinary diagnostic lab. That was a really interesting setting, the Hawke's Bay. The humans were the sentinels for animals, and you are allowed to say to me, well, no surprises if you like, in a way, but we started to anecdotally hear about a lot, say, of redwater in sheep, about cases in farmers, more cases in Pākehā than Māori, more cases in farmers than meat workers, and animals with lepto. But of course there's a natural lag between the reporting of the animal cases compared to the human cases. When we look at healthy animals, we're seeing about every second animal has a titer. So we feel that this is Harjo and Pomona in beef and sheep herds in New Zealand. Every second animal, 99% of herds and flocks have lepto. It's endemic. We think it's maintained, and therefore it's out of kilter with the extreme stress and the flooding. But yeah, that was a surprise. Although interestingly, looking back at that one, now that we know there were so many animals unwell, I guess it wasn't a surprise. Sorry, does that answer your question? I've lost my train of thought a bit, Jackie.
No, it does. The shift. So that's super interesting.
Yeah.
Are there any more questions for Jackie? No? Well, I just want to thank you so much for just a fascinating talk. Before we congratulate her, and I will give her a present, also want to thank everybody here for attending this lecture. And of course, we're very super lucky to have Jackie here for this week, and actually also married up with Isby in the Sydney Conference Centre in the city for next week as well. So we've been able to learn lots from you over an extended period of time. So if I could ask you to thank Jackie in the usual kind of way.